All right. Well, if you're watching this archive, we have had our live session, but I'm re-recording um because the recording did not happen, and that's okay. So I'm going to make sure that you guys who are watching get the same information that we had in our live session. So, the chapters that we covered um throughout the last 4 weeks, we had chapters 1, Intro to Micro, Science of Micro, the Microbiology Lab, Prokaryotes, Eukaryotes, Empire Viruses, Parasites and Vectors, Mycology, Microbial Viability, Growth, and Microbial Genetics, as well as Mutations, Microbial Disease, Transmission. Now, earlier we did have a Kahoot, which I will be sharing in the archive for you all, and we also had poll questions. So for you all, I'm going to kind of show you the poll questions that they were asked so that you get the same questions that they did. Now, the last day of the module is September 20th, 2025 at 11:59. So you want to make sure that you have your final done and complete so that you can just go into the break stress free. With that, we're in midterms and we're moving right along. If you're missing anything this week, make sure that you complete it. Those of you who have to do the discussion forums, make sure that your initial post is this Friday and your peer response, at least one, is submitted by Sunday at 11:59 for full credit. And do not wait to the last minute to get that midterm in. All right, set yourself up for success. You don't want to, wait to the last minute and then technology happens, i.e., I'm re-recording that I am re-recording after the live session because it happens. All right. Now, we went over certain things to kind of go look at. All right. We're going to start with a few tricky areas from past midterms where students got or lost a few points. Okay. So that you, as you're watching this archive, can kind of know where to guide your studying. Now, some students confuse bacteria with viruses. Remember that they are fundamentally different in structure and in treatment. Bacteria are free living cellular. Um, they have walls with metabolic activity. They're treatable with antibiotics. Viruses are acellular, the nucleic acid enclosed in protein. What they do is they have to go in and hijack host cells, and they are not affected by antibiotics whatsoever. Now, the textbook stresses the importance of just recognizing these differences in selecting sterilization, your PPE, and infection prevention strategies. Another common slip that kind of that I saw was forgetting that fungi are eukaryotic, not prokaryotic like bacteria. Now, the book clearly groups fungi under eukarya, and it highlights their true cellular complexity. They have a nucleus, they have organelles. And compared to bacteria, that's a simpler prokaryotic structure. The distinction affects antifungal drug choices, and it highlights why fungal infections in surgical patients demand different types of protocols. On the lab side, pay attention to cultural methods and biosafety levels. Know when and why each is used. Now, in discussing the microbial lab, the chapter or the book emphasizes choosing appropriate culture media, selective, differential, and aerobic. And that's for isolating pathogens, and it explains how labs assign different biosafety levels. Microbial genetics is another pitfall. So you don't want to mix up mutations, plasmids, and resistant mechanisms. Now, the textbook outlines mutations, random changes in DNA that may confer survival advantages. Plasmids are extra chromial DNA that circles that can actually transfer resistance between genes between bacteria. And then your resistant mechanisms, such as enzymatic breakdown of antibiotics or efflux pumps. The chapter the chapters in the book explain how understanding these concepts is going to be really important in infection control and antibiotic stewardship in surgical settings, especially in preventing surgical site infections. Finally, do not forget how microbes are transmitted, especially knowing the difference between your vectors, your fomites, and your aerosols. The book discusses modes of disease transmission that's relevant to our setting in the operating room. Remember that vectors are living organisms like your mosquitoes or your ticks that carry pathogens. Your fomites are inanimate objects like your instruments, our linen, our gowns, and they are capable of transferring microbes. And your aerosols, they consist of tiny particles suspended in air, your droplet nuclei. Now, proper sterilization, handling of material, and control of aerosol generating procedures are stressed as central prevention tactics. Now, each point mirrors the clear, clinically focused framing that the book uses, connecting microbiology to surgical technologists and our roles. So keep that in mind as you're going through your studies. These aren't here to scare you, okay? Like, oh my gosh, if I see this, I'm going to do bad. No, 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 no. It's just to kind of help you to think clear when it comes to the exam, where you can now sharpen those spots and it becomes like second nature during the test. So here's another quick recap. Know your lab gear, your PPE, your safety protocols, especially biosafety levels. We talked about it just last slide. Review the cell structures that separate these two worlds. It will help you categorize diseases and treatment quickly when you talk about prokaryotic and eukaryotic cells. You have your viruses and genetics. What makes viruses unique? They don't have a cell wall, they have to have a host. These are going to be big midterm themes that you're going to see. Understand how mutations and genetic transfers play into resistance and infection risk. The book discusses key genetic mechanisms. Mutations can alter microbial traits. Plasmids enable horizontal gene transfer, often of resistant genes. There's resistant mechanisms like enzymes. And then you look at infection risks and what influences infection control and antibiotic stewardship in the operating room. Don't just memorize. I want you to think about what conditions help hinder microbial growth. The book details factors affecting microbial viability, temperature, nutrients, moisture, oxygen requirements. The book also covers culturing methods and media types. Okay. Be crystal clear on how infections spread, especially in surgical environments. You have transmissions modes are really explained, including direct contact, droplet versus airborne, and even fomite routes. Know the major players and the body systems they affect. Also, vectors versus hosts are huge. The book chapters on parasite and vectors describe protozoa and helminthids, how they survive in hosts, how vectors transmit disease, and what systems are typically affected. The Mycology section covers fungal characteristics, opportunistic infections, yeast versus mold distinctions, and antifungal resistance, especially important for our immunocompromised surgical patients. Now, this is your checklist. If you can confidently kind of talk through each category, you're in great shape for the midterm. Now, like I mentioned, we went through the Kahoot. There are 5 true and false questions, and there are 10 multiple choice questions. And the way that this is set up is for you to kind of trust your instincts and pick. What do I mean by that? When you open up the Kahoot, you'll see that there are, with your true and false, you have 5 seconds to pick. And with your multiple choice, you have 10. And this is all designed to kind of help you to not second guess yourself. So when you have the opportunity, make sure you try this Kahoot out and use it as a resource to help you study for this upcoming midterm. We also had poll questions. So, what I'm going to do now is for the next 5 minutes, I am going to show you the questions that everyone in the live session saw. So, you have the same opportunity to see the questions that they were presented. You'll also know that these concepts are sprinkled throughout the review that we just went over. You'll have the two highly chosen question or options and the questions that went with it. So go ahead, take your time, and I'll be back in 5 minutes. All right. So you have had the same opportunity that those in the live session had. So again, use these as resources as you gear up for your midterm. Now, these are not the questions that you're going to see, but these are concepts that are going to kind of help you. So no matter how you see the question, you're going to know the difference between a eukaryote having a nucleus and a prokaryote not. Okay. Now, with that, this is the wrap up. Okay. Now, in the discussion forum, these are the questions. Why is it important to study microorganisms in everyday life? What are some ways scientists can tell the difference between bacteria, viruses, and fungi? And how do microbes grow and spread? And why does that even matter to us? If you have any questions or any concerns, do not hesitate to reach out. I am here for you. With that, I do want you to have a wonderful rest of your evening. Have a wonderful rest of your weekend. Be safe. You are going to do good on your midterm. I believe in you. And I look forward to seeing everyone who can attend our next live session. Good night, everyone.